Good afternoon, everybody, and welcome to today's ESDR Kitchen live webinar. Just before we get into the main course of the day, I'll just give you a little bit of background for those of you who may be new to the Kitchen series. As you are very well aware, with the COVID-19 pandemic, we are unable to run face-to-face meetings, and the ESDR felt it very important to continue to educate our membership and to have scientific discourse with our membership. And back in July of last year, we set up this series of live webinars, which we called the ESDR Kitchen, which have 4 themes. The first one is the recipe book, which is around research techniques explained. Second is sweet and sour, where we have 2 experts debating or discussing a hot topic in dermatology or skin biology. Freshly baked are really literally hot off the press, newly published papers, which are discussed by the lead authors. And then the 4th theme is molecular cuisine, where an eminent senior investigator presents his or her journey from discovery through their life program of research in their career. But today's webinar is the recipe book and presented by Andrew Ji. And I'm going to hand over to my 2 fellow ESDR board members, Enikö Sonkoly and Julien Seneschal, to take us through to the questions.
Thank you very much, Chris. You are all welcome to this episode. This will be the recipe book again. We are going to hear a very interesting recipe today, an exciting recipe about epigenomic profiling. This is a method called perturb-ATAC, combining CRISPR methods with chromatin accessibility analysis. So we are all very excited to hear about this, and Andrew Ji is going to introduce to us this method and make it simple for us. Now, Andrew Ji is a postdoc at the Paul Kawari lab at Stanford. And his main interest is squamous cell carcinoma of the skin, and he has published some very nice papers about it, especially the recent paper I really can recommend. But today he's going to talk about epigenomic profiling, which is a very powerful method in epidermal differentiation and gene regulation during epidermal differentiation. Now, I can also say that Andrew Ji is going to start his new group at Mount Sinai at New York City. So with this in advance, we can congratulate to him. So he is going to present this in a recorded presentation, but he is going to be here with us for the discussion. It is a very early hour at Stanford, which is why we do like this. But I would like to ask you, all of you who listen, ask your questions. You can send your questions in the chat function or in the Q&A. So I would like to ask Thomas to start the presentation of Andrew Ji.
Hi, everyone. My name is Andrew Ji, and I'm a postdoc in Dr. Paul Kawari's lab at Stanford. I'm also a clinical instructor of dermatology, so I spend half a day per week seeing general dermatology patients and the rest of my time in the lab. So I'm a physician scientist, and my focus has been predominantly on studying skin cancer, particularly squamous cell carcinoma. But during my postdoc, I also had the good fortune of being able to help out with a method that our lab developed called perturb-ATAC, which will be the focus of my talk today. So this is a method that allows for simultaneous detection of CRISPR guides and open chromatin profiling in the same single cells, which we think is a very useful approach for trying to understand gene regulation and how it occurs in the skin. So I want to thank the organizers for this invitation. I'm very excited to speak with you all. I have no conflicts to disclose. Before I even begin, I want to first start by acknowledging the amazing team behind perturb-ATAC, which included Adam Rubin, a former graduate student in our lab, as well as members of Dr. Howard Chang's lab, including Kevin Parker, a graduate student, and Ansu Satpathy, a former postdoc in Dr. Chang's lab. So for many years now, our lab has been very interested in understanding how different cell types can carry out different functions, despite the fact that for the most part, they all use the same set of DNA instructions. And so a large determinant of this is, of course, the specific genes or combination of genes that are expressed within these cells. But our question is, how does this happen? How do cells select which genes are expressed in certain tissues? If we zoom in a little bit into a single strand of DNA, in a very oversimplified scenario, what determines the activity of a gene, whether it is off or on, comes down to a transcription factor binding to a specific sequence near the gene's promoter, called an enhancer, that loops to and physically contacts the promoter, recruits transcriptional machinery, ultimately to drive expression of the gene. But of course, this is oversimplified because DNA isn't just floating around as these single strands. If we zoom out just a little, we can see that in order for this particular transcription factor to bind to an enhancer, the enhancer must be physically accessible. So how do we know if an area of the genome is accessible? One method to do this is the assay for transposase accessible chromatin with sequencing, which is better known by its acronym ATAC-seq. How does ATAC-seq work? It takes advantage of a hyperactive mutant TN5 transposase that selectively integrates DNA sequencing adapters into regions of DNA that are more open. So visualizing an example of data coming from ATAC-seq, these 6 tracks you see that are stacked on top of one another, here have the genome locus depicted on the x-axis and ATAC signal on the y-axis. So areas with high signal are referred to as peaks, which correspond to enhancers and promoters and are highlighted in the light blue rectangles you see. What you may also notice here is that each cell type has a distinct profile of where its peaks are. And so you can imagine capturing a profile like this across the entire genome, genome-wide, would give you the epigenome, or a readout of any particular cell state. So the question we have is, what factors are important for establishing the epigenome and therefore the state of any given cell? So one powerful approach to do this is through CRISPR screens, where you can perturb many such factors, and determine functional outcomes. So at this point, I want to make a distinction between a traditional screen and a single cell perturbational screen such as perturb-ATAC. So in a traditional screen, the phenotype that you read out is actually quite simple, just growth or death. But in a single cell perturbational screen such as perturb-ATAC, you can set up the screen in the exact same way, transducing a library into a pool of cells. But instead of a simple readout, like growth or death, your readout now becomes an entire ATAC-seq profile. So perturb-ATAC is one form of what some people refer to as a high content screen, where your phenotype readout is higher content than a traditional screen. So how does this work? The basis really for this is the ability to simultaneously detect a CRISPR guide and perform ATAC sequencing from the same single cells. And so when we developed this approach, we used the Fluidigm C1 microfluidic chip platform that isolates individual cells within wells. But it's important to note here that the platforms that existed when we developed this are now, such as the Fluidigm chip, are perhaps a bit obsolete at the moment, because I want to highlight additional other methods that are a little more high throughput these days, such as combinatorial indexing or droplet-based microfluidics that allow for much higher throughput. But again, they all require the same principle of being able to detect the guide and perform ATAC simultaneously. So in our method, each single cell is captured into these wells. TN5 transposition occurs in these wells. It is performed, and then guides are actually detected through targeted primers. Then both sets of fragments are amplified, indexed, and ultimately sequenced together. So to validate this approach for guide detection, we first performed a simple mixing experiment where we transduced 2 different guides into 2 separate pools of lymphoblastoid cells, in this case, the GM12878 cell line, and then mixed them together. And so here on the right, we could see that we could readily detect each guide distinctly in single cells. So the next question was whether the associated ATAC-seq profiles from single cells reflected the expected chromatin changes in the perturbational scenario. So first, performing a CRISPR-I experiment in bulk cells, we found that upon transduction of a guide to SPI1, which is a transcription factor involved in B cell development, the peak associated with the SPI1 promoter was lost, as you look at these red tracks for the bulk scenario. And we can see that in the perturb-ATAC format at single cell level, we again see that upon CRISPR-I of SPI1, we also abolish the peak associated with the SPI1 promoter. And you can aggregate global genome SPI1 motif accessibility at all genome regions across the entire genome that contain a motif for SPI1, and see that the presence of the SPI1 guide globally decreases accessibility at sites that contain the motif at both bulk and single cell levels. So the red squares on this plot on the right show the bulk data, while the blue circles show the perturb-ATAC data, which matches up nicely with the bulk data. So with confidence that we could consistently detect guides and perform ATAC-seq from the same single cells, we could start to think about designing both screens as well as different analyses to gain insight into how particular factors may control cell state. So at this point, I'd like to introduce one analysis approach on how to infer regulatory networks from perturb-ATAC data. So it relies on the powerful ability of single cell analysis to detect heterogeneity across cells at baseline. So for example, if you look at the top rows of idealized data, if we observe a correlation between the accessibility of two different motifs, and when you perturb a certain factor, and you observe that that correlation is lost, you can start to infer that the perturbed factor is involved in regulating the relationship between these two motifs, either by working together where one partners with the other to drive the accessibility of the third, or where the perturbed factor can independently regulate both factors. And so in the bottom row is true data coming from one of our perturb-ATAC experiments with the GM12878 cell line. And what we see is, in this case, we observe a strong negative correlation between the BAT-F and TRIM22 motifs across wild-type single cells, implying a potential negative regulatory relationship. And then if you look more broadly across pairs, so once you perturb DNMT3A with CRISPR-I, what you notice is that that negative correlation relationship is lost. And so you can not only just look at two pairs, but you can look more widely across other pairs broadly across the genome and find instances of where a high correlation at baseline is lost upon perturbation of DNMT3A, which is shown in the heat map correlation plot on the right, where the bottom rows show baseline correlation between pairwise between motifs, and the top rows and the top half shows the DNMT3A CRISPR-I cells, where upon DNMT3A, which is a DNA methyltransferase, upon inhibition of this particular gene, you see a loss of that correlation. Okay, so how do we leverage this method to learn more about the skin? Our lab has long been interested in how transcription factors regulate the process of epidermal differentiation, whereby a single layer of progenitor keratinocytes can give rise to stratified differentiating cells that form a functional barrier that protects us. And this is a highly dynamic process involving the suppression and induction of thousands of genes in a highly coordinated manner. So like many other labs, we've used an in vitro differentiation approach whereby we add calcium to primary keratinocyte progenitors cultured. And over the course of 6 days, these cells differentiate and start to express many of the same genes that are present in the suprabasal stratifying differentiating layers of the upper layers of the normal skin tissue. So if you perform single cell ATAC-seq on cells harvested at these 3 different time points, day 0, day 3, and day 6 of this in vitro differentiation process, you can perform dimensionality reduction with t-SNE, and you can broadly observe that these cells will tend to cluster according to their day of harvest, as shown in this t-SNE plot. But there are some cells that cluster with other groups, suggesting that this really is a continuous process. For example, some day 6 cells cluster more with day 3 cells, suggesting they might be more stragglers in the differentiation process. And you can also perform unbiased analysis, such as pseudotime trajectory analysis, where you assign pseudotime values to each individual cell, again reflecting the continuous nature of differentiation, but still corresponds nicely to their day of harvest. So through the pseudotime analysis, we recovered 3 modules of dynamic motif accessibility across pseudotime that either become that are open to begin with and become closed over pseudotime, such as those in module 1, ones that are closed to begin with, open in the middle of pseudotime, and then close again, such as in module 2, and then finally module 3, which is closed to begin with, and then of course opens later pseudotime. And some specific transcription factor motif quantifications are shown on these right set of t-SNE plots, where you can see that the transcription factor Jun is highest early in pseudotime. Conversely, the transcription factor motif EHF is most accessible later in pseudotime. And of course, there are also the 3 on the bottom that are part of module 2, where they are highest in accessibility in the middle of pseudotime. So with these factors in mind, we set up a perturb-ATAC screen to ask what happens to the epigenome keratinocytes upon perturbation of these factors. So we transduced guides targeting factors that I just showed you from these 3 different modules, allowed the cells to differentiate for about 3 days, and perform perturb-ATAC. And on the right, you can see the detection of these 5 guides along with their non-targeting control guides across the single cells. And on the top is the situation where each cell received 1 guide, so single cell knockouts on the top. But we also took the opportunity to generate double knockout cells where we could transduce pairwise combination of these guides across single cells. And that's shown in the heat map on the bottom. So because we had the pseudotime trajectory from wild type cells, we could assign all perturbed cells a pseudotime score and compare how specific perturbations shifted their differentiation states. So in the top heat map here, we binned wild type cells according to their pseudotime scores. And we see that across these 8 pseudotime bins, the day 0, day 3, and day 6 cells occupied expected bins across pseudotime. But if we look at the bottom heat map, we see that when we scored the perturbed cells for the same pseudotime values and show the proportion here of how each perturbational cell type is binned into these pseudotime bins, we find that for transcription factors like CEBPA and EHF that are known to be involved in epidermal differentiation, we find that upon perturbation, these cells tend to pause or be enriched early in pseudotime, suggesting that they're failing to differentiate. Meanwhile, a factor like Jun-B upon perturbation, AP1 factors are known to maintain the progenitor keratinocyte, and upon knockout, these cells tend to shift later towards differentiation, suggesting that they are now differentiating. And you can also ask specific questions about how perturbations affect specific motif accessibility. So in this particular heat map, the rows represent the perturbational identities of cells, while the columns are specific motif accessibilities, genome-wide relative to the non-targeting cells. To highlight a few examples here, we see that the first 2 columns, which are ETS motifs, both decrease in accessibility upon knockout of EHF. Similarly, AP1 motif accessibility go down broadly upon knockout of Jun-B. And consistent with this as well, CEBP motifs also decrease upon CEBPA knockout. So you can also look for other transcription factors that are known to be involved in epidermal homeostasis or differentiation, like NF-kappa B or P63. And you can observe that specific perturbations can either open or close these sites, again, going back to the idea that we can start to infer regulatory relationships across the genome. Because we're able to generate double knockout cells, this enabled a deeper look into potential epistatic relationships between pairs of transcription factors by comparing single to double knockout cells. And so we categorize genetic interactions into either synergy, buffering, or additive relationships, where synergy was defined as a positive relationship, defined as an observed effect on accessibility that was greater than expected. Buffering, which is a negative interaction, was defined as an observed effect lower than expected, while an additive relationship reflected the expected effect. And as we look across all of our double knockout cells, we can calculate the proportion of these interactions across all motif accessibility regions. And these bar plots on the right just show the proportion of synergy, buffering, and additive interactions in each double knockout condition. And we can see there's quite a bit of variability in the proportions of these genetic interactions. We think this is something quite interesting and worth following up on. And the timing of the seminar is quite nice because there was a recent methods primer paper published in Nature Reviews Methods Primer. It's a very comprehensive look into chromatin accessibility profiling methods, touches upon other assays other than ATAC-seq, as well as perturbational approaches. So I'd highly recommend reading this for a very detailed look at these methods. I want to thank all members of the Kavari and Chang labs for their hard work on this project. And I'll finish with a bit of shameless self-plugging here. I'm very excited to announce that I recently accepted a faculty position at Mount Sinai School of Medicine in New York City to start my own lab this coming fall. So I'll be a member of the Department of Dermatology, the Black Family Stem Cell Institute, and the Tisch Cancer Institute. And I didn't get a chance to talk about my main project for my postdoc, which involved single cell and spatial profiling of patient samples of cutaneous squamous cell carcinoma. But this work was published last summer, so have a look if you're interested. And we'll be looking for highly motivated young scientists like yourself to use these approaches to better understand how different cell types communicate with one another in the tumor microenvironment, as well as within other skin diseases. For those of you that are not so familiar with New York City, this is a photo of Central Park looking downtown or southward. You can see the Empire State in the background here, Empire State Building in the background. And Mount Sinai is actually in a wonderful location right bordering on Central Park. And it's probably just out of the photo here, maybe around here. So it really is a nice place. And I'm very excited about the potential synergy and excitement about starting a lab there. And so any of what I've spoken about today or about skin cancer piques your interest, please do not hesitate to reach out to me. I don't yet have a Sinai email address, but my Stanford one should be active for quite a while since I don't start until September. So thank you all for listening, and I'm happy to take questions.
So we are back, and thank you, Andrew, to be here on live at very early time for you. And thanks a lot for this great presentation and very clear, and it was very instructive. So we have some first question coming on the chat, so you can also ask a question. So the first one is from Daniel Kaufman. So have you checked dermoid cyst with this method? The cyst just multiple tissue types in the same cyst, and maybe your method could identify the problem.
Yeah, thank you, Daniel. That's a great question. So we have not looked at any intact tissue samples with this technique. So far, it's only been done from cells that have been cultured, mainly because it's more feasible to perform CRISPR screens in cells that are coming off plates, as opposed to being able to transduce guides and make sure that the tissue has Cas9 expressed. So it becomes very difficult experimentally to try to set up this type of approach in intact tissue, but definitely it would be fantastic, or it'd be very interesting to profile either through single cell ATAC or other chromatin profiling methods to dissociate cells out of something like a dermoid cyst to profile it, but we've not done that.
Thank you.
Enikö? Yeah, thank you, Andrew, for this really nice presentation and very pedagogic. Well, I don't see now any more questions. Please send your questions. Meanwhile, I can ask, it's quite interesting. You looked at these interactions between different transcription factors during epidermal differentiation with this method. So did you find some surprises, or was everything as you expected?
It's a great question. So whenever you generate these high dimensional data sets, you definitely want to see things that you expect that are that give you confidence that the method is working from a technical standpoint. So you're right that many of the factors that we perturbed were known or had known roles in epidermal differentiation. So many of the things that we highlighted were expected. There were definitely less, I think, in terms of the multiplex knockout. You know, there aren't many data sets at all that examine this. And one thing that I didn't mention was that for some of the synergistic interactions, what we did observe was that there was co-localization of motifs at the specific sites that were sort of opening or closing in a synergistic fashion. So that would be one model to explain the synergy that you're seeing, is that there's co-occupancy of these TFs at specific sites in the genome.
Yeah, thank you very much. Because following this question, because you have done some beautiful works to show the landscape of squamous cell carcinoma, and do you think that your technique could be applied to decipher the different step of the development of squamous cell carcinoma, as example coming from solar lesions, actinic keratosis, and then true squamous cell carcinoma? What do you think?
Yeah, great question. So definitely this could be applied in a tumor initiation scenario that you're describing. Most of the time, or I would say the best application for this type of method in asking a question like that is with a good model for tumor initiation. So whether it's a mouse model or xena, probably the most feasible approach would be through either a xenograft model, where you can take cancer cell lines or human cells in a dish and manipulate them prior to injecting them into the mouse. Or we have in the lab, we use some organoid models where we can reconstruct invasive tissue, three-dimensional invasive tissue in vitro. And so all of those scenarios would be great scenarios to try to use the system.
And so just another question, but for people who are not really aware of this technique, but what for you are the main limitations of this technique? What we should be aware? Is there some risk of experimental noise due to the data generated or something like that? What are they?
Great question. Yeah, so definitely all single cell data sets are inherently pretty noisy to begin with. This particular method, I would say the main limitations I alluded to this, but the throughput is definitely lower than what's available now. So the Fluidigm chip is quite, in many ways, obsolete. For this approach, for this method, it was a couple hundred cells per experiment, and now you're getting an order of magnitude a couple thousand cells, really. So definitely throughput is a limitation, and it depends on what questions you have and whether you can answer the questions through a screen with a couple hundred cells as opposed to the ones now that are really doing thousands. Data analysis is not necessarily straightforward a lot of the times. You do have to make sure there's some good quality control before you sequence, there are definitely benchmarks to hit before you sort of dive in. And the reason why I brought up that methods primer is because they do a really wonderful job of sort of outlining all the different quality control parameters that you want to be aware of, as well as some approaches for making sure the analysis is sound and making sure that you know, it isn't, the signal to noise ratio is good. And so there are definitely things that you want to ensure before you sort of you know, dive in.
Well, I have also a very general question, just quickly. I mean, how should we think about these open chromatin regions? So I mean, are they always enhancers, promoters, or do you find open chromatin at places where you wouldn't think? Do you have to validate this approach somehow?
Yeah, so then that's a great point, because ATAC-seq is really one method and among a compendium of methods. And to really get a solid view of the genome, you do have to look at other things, whether it's ChIP-seq or other approaches, to make sure, because you are inferring a lot of things from ATAC sequencing data, and it's really just a view of the physical, you know, structure in a way of the chromatin. You can't determine function. You're really just inferring that, especially looking at, you know, even though you're looking at transcription factor motif accessibility, you don't really know if the transcription factor is actually there. All right, so you're making some assumptions. So it does require, if you do focus in on a specific factor that you think is doing something, you do have to validate with more targeted approaches and make sure that it's physically binding chromatin, or that it's actually driving expression. So a lot of times people combine this with transcriptomics to really assess how gene regulation is occurring.
Yeah, makes sense.
So thank you very much, Andrew. It was a great pleasure to have you on this recipe book ESDR Kitchen webinar. And it's time to conclude, and we let Chris Griffiths to announce the next one. Thank you very much.
Thank you all
so much, Julien. Thank you so much, Enikö. And thank you again, Andrew, and all the best in your move across country to New York City. I'm sure we'll be hearing more great stuff from your own lab. So to move on to our next kitchen theme, which will be on the 10th of February. So put that in your calendar. And we've got two of our freshly baked, hot off the press news items. First will be presented by Gary Reynolds from Newcastle, talking about developmental cell programs in inflammatory skin disease. And the other presentation will be by Sally Dabelstein from Copenhagen, talking about the functions of glycans in human epithelia. And so with that, thank you so much for joining, and enjoy the rest of your day or evening or wherever it is around the world. And I hope you enjoyed it. We're always looking for feedback, and take care. Bye.